Princeton High School Academic and Career Hall of Fame was established in February of 2017 to recognize distinguished alumni who have demonstrated outstanding achievement in areas such as citizenship, leadership, academic endeavors, and career achievement. The goal is to recognize Princeton High School alumni that have distinguished themselves by their contribution in their field of work or study to the Princeton High School community and to society as a whole. Dr. Rich Hallberg graduated from Princeton High School in 1983. After high school, he received his Bachelor of Science Degree with majors in Biology and Chemistry with a minor in Mathematics from the University of Iowa. Dr. Halberg also graduated with a Ph.D. from Michigan State University in Biochemistry. He is currently a professor in the Departments of Medicine and Oncology at the University of Wisconsin with appointments at the Carbone Cancer Center, Institute of Clinical and Translational Research, and the William S. Middelton Veterans Hospital. In the fall of 2021, he was appointed as Senior Director of Biomedical Research Facilities, overseeing 34 cores in the School of Medicine and Public Health. Dr. Halberg proved a long-standing theory in cancer biology was wrong. Scientists believe for over 100 years that each cancer is derived from a single rogue cell. He and his research team demonstrated that cancer can be derived from multiple rogue cells, and that is why cancer can be so difficult to treat. Dr. Halberg's research has been described in over 70 publications in leading scientific journals and supported by grants from the American Cancer Society, American Association of Cancer Research, Department of Defense, National Cancer Institute, and NASA. Please join me in welcoming and honoring Dr. Rich Hallberg.
Unfortunately, you're not that lucky. I got plenty to say. So I'll try to make it quick so we can go watch Princeton beat Hall tonight. Okay? So any one of you could be standing here someday. My beginnings were probably very similar to almost everybody in this auditorium. So I started at Douglas Elementary School. How many people went to Douglas? I guess you all do now, right? Because it's all back in the day, it was just kindergarten through 1 through 4. So went to Douglas, then Washington Middle School, Logan, and then Princeton High School. So I spent all my early years in Princeton. And I had many wonderful teachers. They were asking me today who was my favorite high school teacher. He's standing in the back, Mr. Young. So, but the teacher that probably had the biggest effect on me, and that was because she affected my life so early, was a woman by the name of Susan Baker. She was my 4th grade teacher, and the amazing thing about her is she taught me that learning was fun. And I enjoyed every day of class. I enjoyed her class so much is when I went to 5th grade in Washington, I would ride my bike back to Douglas every day to see her. So she was a phenomenal teacher. And it's people like that that really change people's lives. So all the teachers are here deserve a Did anybody have Miss Baisley, or are they related to her? I know she has family. So she was an amazing teacher. And like you, I probably did a lot of the similar jobs that she did. So how many did tacit corner? Now, do you guys get right on the machines? Yeah, see, Uncle Gary and I didn't write on machines. They didn't have machines back then. We had to reach up and pull them down. Worked at LCN. Uncle Gary also worked at my uncle's here. They also worked at LCN. I did 2 years. I think he did 40 years. And then I worked at Wyatt Hills Golf Course. So I worked at many of the places that you did. And the difference really came in when I went to college. So I had the opportunity to go to college. I had great scholarships from different places in Princeton and different universities. So I ended attending 2 or 3 of the Big Ten schools. So I went to the University of Iowa and then Michigan State, and then I ended up at Wisconsin. Now that's when the Big Ten was actually still just 10 schools. And my dad would often say to me, son, they're adding schools faster than you're finishing them. But the key piece of advice that my dad gave to me was that whatever you do, I don't care what you do, whatever you do, make sure you love it, because you have to do it for the next 4 years. And that's, any of you that take that to heart, if you love what you do, your chances of standing here are really, really, really good. Because if you love your career, it's not work. It's just fun. And I get to have fun every single day. So I'm a professor at the UW, as they said. And you guys know what a professor does? No? Guess what a professor does? So yell out a couple different things. What do you think I do every day? Eat. I heard eat. How many people think I teach like 3 classes, 400 hours a day? Yeah, no, I don't teach ever. I teach 10 lectures a year. So I don't teach very much. I teach cancer biology. Primarily what I do is research, and in order to do that research, I have to get funding for external sources. So you heard that some of my fundings come from the Department of Defense, or NASA, or the National Cancer Institutes. And I'm just going to tell you 2 stories about 2 different projects that I've done over the last 25 years that have made a little bit of difference in maybe some people's lives in this very room. So in order to do that, though, I'm going to play this video. This video comes from a movie some of you may be familiar with. It's called Jurassic Park. And this is Mr. DNA. So in order for me to talk to you about the work we did with DNA, I thought this was a great way to introduce the idea of DNA.
Where do you get 101-year-old dinosaur? Shh.
What? Oh, good. Mr. DNA, where did you come from? From your blood. Just one drop of your blood contains billions of strands of DNA, the building blocks of life. A DNA strand, like me, is a blueprint for building a living thing. And sometimes animals that went extinct millions of years ago, like dinosaurs, left their blueprints behind for us to find. We just had to know where to look. 100 million years ago, there were mosquitoes, just like today. And just like today, they fed on the blood of animals, even dinosaurs. Sometimes, after biting a dinosaur, the mosquito would land on the branch of a tree and get stuck in the sap. After a long time, the tree sap would get hard and become fossilized, just like a dinosaur bone, preserving the mosquito inside. This fossilized tree sap, which we call amber, waited for millions of years with the mosquito inside, until Jurassic Park scientists came along. Using sophisticated techniques, they extract the preserved blood from the mosquito and, bingo, dino DNA. A full DNA strand contains 3 billion genetic codes. If we look at strains like these once a second for 8 hours a day, it'd take 2 years to look at the entire DNA strand. It's that long. It's gonna hold. Now that's where our geneticists take over. Thinking machine supercomputers and gene sequencers break down the strand in minutes. And virtual reality displays show our geneticists the gaps in the DNA sequence. We use the complete DNA of a frog to fill in the holes and complete the code. And now we can make a baby dinosaur.
Taking DNA from woolly mammoth and working with elephant DNA and trying to bring back the woolly mammoth. That's happening. So do you think I made a dinosaur in my lab at the University of Wisconsin? No, because if I would have, I probably got this award a couple years ago. So no, I haven't done that. But I've done something different. And what I did is I took little pieces of DNA, not from a frog, but from a jellyfish. So this is a jellyfish here, and that jellyfish has a protein in it called green fluorescent protein, and when you shine light on it, it fluoresces green. And so you can see that. And down here is a coral, and it has a red fluorescent protein in it. The fluorescence is red. And so what I did is I took the genes encoding green fluorescent protein and the gene encoding red fluorescent protein, and I took it from the coral, the jellyfish, and I put it in the mouse. So this mouse now has DNA from the jellyfish and the coral. So why do you think I did that? Anybody have any idea why Rich Hallberg, who graduated in 1983, would do that? I saw one hand. Don't want to say? Any guesses? Just for fun? Designer animals? You guys can get a mouse that fluoresces green. The reason I did it is I wanted to understand where tumors came from. So the long-standing theory in cancer biology was that every tumor was derived from just one cell. You had one cell in your body that went bad, and that tumor or that cancer formed from that one bad cell. And so I wanted to test that hypothesis, so I made that mouse. And so when you look at this mouse with these DNA from the jellyfish and the coral, and you look at the intestine of that mouse, you can see some of the cells fluoresce red, and some of the cells fluoresce green. And when you look at the tumors that form in the colon of that animal, you can see that the tumors are mixtures of red and green cells. They're not all red, and they're not all green. So that proves that long-standing hypothesis wrong, that it's not just a single cell. In this case, it had to be at least one green cell, and it had to be at least one red cell. So why is that important? We need to understand the origin of the cancers in order to better treat them. Because if I design a treatment to cure a cancer, and I target just the green cells, the red cells will survive. The cancer will still be there. Vice versa, if I design a drug that targets the red cells, the green cells will still be there. And in fact, these little devils work with each other. So if I design a drug that targets the green cells, sometimes the red cells say, no, I'm going to protect you from that drug. So we really need to understand the tumor, make up the tumor in order to more accurately treat the cancers. And you can see in these mice that you can get all kinds of variations in the pattern of the tumors. And in fact, we don't even think it's just two different cells that have gone bad that give rise to a tumor. We think it may be as many as 6 or 10 different cells that come together to form these tumors. And that's what makes them so difficult to treat sometimes. All right, guys, any questions about that? So why do we care about this? Well, we care about it because in the United States, every year there's about 2 million new cases of cancer. And you can see it's all over the United States. It's not just in any one given place. So it's not only we worry about cancer in patients that live in the United States, it's all over the world. And it's even when people leave Earth and go to other planets. So I got one more video for you. So has anybody seen The Martian? Raise your hand. You've seen The Martian. Good movie. It's got one bad word in it, so close your ears when that happens. Okay? I don't want to get fired. This is my first day on the job.
I guarantee you that at some point, everything's going to go south on you.
Really?
And you're going to say, this is it. This is how I end.
Martin's dead. We have to go, man.
Now you can either accept that, or you can get to work.
This will come as quite a shock to my crewmates, and to NASA, and to the entire world. But I'm still alive.
Surprise.
Here's the rub. It's gonna be 4 years for another mission to reach me.
4 years.
And I'm gonna have designed to last 31 days. So I gotta make water and grow food on a planet where nothing grows. But if I can figure out a way to make contact with NASA, none of this matters anyway.
We've got an incoming message.
My God.
space, Neil Armstrong.
Okay, so let's do the math. I have enough food to last for 50 days.
He's going to starve to death long before we can help him.
So I'm gonna have to science the shit out of this.
He's 50 million miles away from home. He's totally alone. What the hell is he thinking right now?
I am the greatest botanist on this planet.
And I'd save my body.
But we need the Hermes crew.
We either have a high chance of killing one or a low chance of killing 6.
I'm not risking their lives. It's bigger than one person.
No. So,
NASA rejected the mission.
So, if we do this,
We're talking mutiny. If anything goes wrong, we die.
do you realize how crazy this is? We had no other option.
No matter what happens, tell the world, tell my family, that I never stop fighting to make it home.
So one of the big challenges is on a trip to Mars, you heard him say, it's going to be 4 years before somebody's going to come and save you. They're going to be on resident on Mars, living on Mars for over a year. So one of the major problems is exposure to radiation. They're going to be exposed to a lot of radiation. In fact, the amount of radiation they're going to get exposed to is about 4,000 chest x-rays worth. That's how much radiation. You maybe say, well, why don't they just put shields on the spaceships or something like that? If they do that, guess how long it takes them to get to Mars? But even longer. So those 4 years now become 20 years. So which of you in the audience is going to be the astronaut that's going to take 20 years to go to Mars and come back? Probably not any of us, right? Oh, you are? Yeah, volunteer, right here. Sign him up, get his name. So the biggest challenge with exposures to that kind of radiation, of course, is there's a great risk of developing cancer. There's also damage to our central nervous system, or the way our brain works, and our cardiovascular system. So there's going to be lots of issues. And so a study that just came out this week actually showed that astronauts that travel in space, even for short periods of time, 1 to 2 weeks, they have a lot of damage to their DNA. Those little DNA molecules we were talking about, taking them from jellyfish and putting them in ice, so on and so forth, they have lots of damage. And you know what? We expected that. And we expected that because about 15 years ago, we did an experiment with a mouse. So our mouse here, our astronaut, he did go to Mars. He actually went to Brookhaven National Labs in New York. I got to go with him. And they have a particle accelerator there. And the mice actually sit in a cage in this room here they call the target room. And they generate the radiation here that simulates space radiation. So what they do is they make radioactive iron and they make radioactive silicone. And they run that through this target room and they hit the animals in these cages. So we're simulating space radiation and we're seeing what damage is done to our mice. So we can predict what's going to happen to astronauts because we don't want any surprises. NASA hates surprises. For everything they design, they have at least 3 different backups for it. So what happens in this case is when the radiation hits the DNA, so this is the DNA molecule here, it causes the DNA to be shorter. And that happens with a specific type of sequence called microsatellite sequences. So these are just DNA sequences that repeat over and over and over again. So it may just be A 60 times. And those long repeat sequences, when they get hit with radiation, instead of being 60 A's, now become 50 A's. So why is that important? So what NASA asked us to do with our mice is could we predict how much damage was being done to a human astronaut when they're in space by tracking how much damage was being done in these mice. And it turns out these repeat sequences, these microsatellite sequences, are much, much more sensitive. So we could very accurately predict how much radiation exposure a mouse had had. So if they sent us a mouse and they said it had been exposed to radiation, and they asked us how much, we could say exactly how much it was based on how much damage we were seeing in these sequences. Now this is where serendipity steps in. So what happened is that tumors, tumors that have short microsatellite sequences, those tumors are really susceptible to a new form of therapy called immunotherapy. And the reason is that the cancer cells, they're really, really, really good at growing, but they're not really, really good at replicating their DNA accurately. So they make lots of mistakes. And so just like with the radiation, those sequences become shorter. And when that happens, they start producing proteins where our body goes, wait a second, I don't recognize that protein. I think I should kill that cell. And so what immunotherapy does, it says, go ahead, kill that cell. You're right, that's a cancer cell. We want that cancer cell out. So we designed a series of experiments for NASA to see whether or not we could predict how much damage was being done to a person's DNA while they were in space. And it turned out that those same sequences are now being used to determine whether a person is eligible for immunotherapy. So a person will come in, they'll have their DNA tested. If they show that these microsatellite sequences are shorter, then they're great candidates for immunotherapy. And they'll start them on immunotherapy. And in some cases, it's actually cured the patients of their cancers. It kills all the cancer cells in the body. So just by chance, we were working on one project and it overlapped with another project. And it has very important consequences for a lot of people. So I told you about two different projects that I worked on for 25 years. That's not the only thing I did. I taught those 10 lectures a year, don't forget that. So I did a few other experiments, and all that time, this person here, his name is Mr. Terry Fritter. At the same time, not only did he take care of all my mice, but he took care of mice for 25 other laboratories. So I often talk to Mr. Fritter, he just retired, and I said to him, I said, thank you so much for taking care of my mice. And I go, you know what, you've done more for science than I have. He goes, how do you figure? You figured out that this way to tell whether people get immunotherapy. What? I don't get it. I said, I answered one question. You helped 25 guys like me answer one question. So all contributions are important. So it doesn't matter what career you choose, just choose the one that you love, okay? That's my one word of advice. And I always end every lecture at the UW with this one quote. It says, dare to live the life that you have dreamed for yourself. Go forward and make a difference. Make those dreams come true. And there was many times in my life that I could have stopped, but I didn't. And there was even the University of Wisconsin. I got rejected there 3 times. And now I'm in charge of one of their programs for $40 million. So go figure, right? So just don't give up. Just keep fighting for your dreams, and they will come true. That's all I have.
Thank you all. Beat Hall. Happy Homestead. You're free to go.